friends, welcome to SAM Biology class. Transcription is a biological process by which RNA is synthesized from a DNA template. The enzyme RNA polymerase, the enzyme RNA polymerase, which is responsible for transcription, was discovered independently in 1960 by Samuel Weiss and Gerald Horowitz This enzyme reads the DNA sequence and transcribes it into RNA. As this enzyme is DNA dependent for RNA synthesis, it is more specifically called DNA dependent or DNA directed RNA polymerase. RNA polymerase is essential to life and is present in all living organisms and some viruses. It couples together ribonucleoside triphosphates on a DNA template. As this enzyme can join two ribonucleoside triphosphates de novo, it do not require a primer to initiate RNA synthesis. By contrast, DNA polymerase requires a RNA primer to initiate replication. In prokaryotes, there is a single RNA polymerase that transcribes all types of genes. Whereas in eukaryotes, there are three different nuclear RNA polymerases that transcribes different sets of RNA. The prokaryotic RNA polymerase, for example, the one which is present in E. coli, is a multi-subunit protein complex. It consists of 5 subunits that include 2 alpha, 1 beta, 1 beta prime and a smaller omega subunit. All these 5 subunits together form the core enzyme or the core polymerase or the core polymerase. Alpha subunits are identical. Each one is 36.5 kilodalton. The subunits are primarily structural and are involved in assembling the holoenzyme. Another subunit is beta, whose molecular weight is 150 kilodalton. It has the polymerase activity that synthesizes RNA. The largest subunit is beta prime. It is 155 kilodalton and it binds to the DNA non-specifically. The smaller subunit is omega, which is also involved in assembling the holoenzyme. It may also help maintaining the structural integrity of the RNA polymerase. The core enzyme is not capable of initiating transcription from a given promoter. In order to initiate promoter-specific transcription, the core enzyme binds with another protein called sigma factor. The core enzyme and the sigma factor together forms The holoenzyme forms the holoenzyme. The holoenzyme is capable of binding to a given promoter and then it initiates RNA synthesis. Sigma factor mediates the binding of RNA polymerase to a given promoter. It is involved only in initiation and it has no role in elongation. After initiation, sigma factor is dissociated and then the core polymerase extends the RNA chain. The core polymerase form a crab class structure with an internal channel running along the full length. All RNA polymerases requires metal cofactors such as zinc and magnesium ions for their activity. So friends, this is all about prokaryotic RNA polymerase. For more updates, please subscribe my YouTube channel Sam Biology Class.